of bugs. That's the Baltimore Underground Science Space. And I'm here in Baltimore, Maryland. And we have folks on the line and also folks in the room. Today we're going to talk about structural color, which is a very interesting phenomenon in nature. And to help us learn more about structural color, we invited Colin Ingham, who is the CEO and founder of a company in the Netherlands called Hoekmine. Hoekmine is not a Dutch word, it's not an English word, it's a mixture of the two. Mine is English for mining from the environment. So, and then hoek is the Dutch word for angle. So, and we're going to learn a little why Dr. Ingham chose to include an angle mine in his presentation. So, in his company's name. So, Dr. Ingham, we're looking forward to hearing from you.
Thank you very much, Peter. It's really appreciated, and I think you've got the perfect audience there. Moving from microbiology and science into art is pretty much what we're going to try and do and talk about here. So I look forward to hearing your questions, and yeah, let's do it. So the title is Using Bacteria as Building Blocks, and that's very deliberate. And so what I'm going to do is talk a little bit about structural color in life and why it's not pigmentation or anything like that, and then why it's important and why it's interesting, and then maybe sort of what we can do with it, maybe commercially, maybe scientifically. So that's kind of pretty much where we're going with this. So the first part. What it is for living systems, and it's really common and really important in life. So all the organisms that I show here are either partially or completely structurally colored. And some of them you're going to know. You're obviously going to know the peacock. And I think most people know that the brilliant colors from the peacock, there isn't actually really a blue or a green pigment there, that if you take a peacock feather and you grind it up really fine, you actually lose the color. And that's not just because you're mixing blue and green pigments, it's just not there. It's more to do with a nanostructure which is interacting with light. And some of these other examples, they're either partially or completely structurally colored. So plants, flowers commonly use structural color. They're kind of a bit complicated because the structural color is often combined with pigments as well. So it can be kind of hard to work out what's going on, but often the two things work together. And you'll also see this sort of thing in butterflies too. Often in the wing of a butterfly, both of these things are happening at the same time. But also in things like certain types of squid, crustacea, or the panel H, which is macroalgae, seaweed, or commonly in birds or in plants, this phenomenon exists. And it's almost always important. Usually, if you're structurally colored, you either want to be seen, either because you want to attract your own species, or maybe you want to warn another species that you're really dangerous. Another possible use is to affect the way organism deals with light. So some plants use structural color and they bounce the light towards their photosynthetic reaction centers in dim light. So it's kind of acting almost like a sort of antennae to harvest light. So obviously it's a visual phenomenon, and obviously most uses are connected with light. You have to be careful though. There are situations probably where organisms appear structurally colored and the structural color is an accident. It's a side effect. So you kind of can't always assume it has a function, but usually it does. But the other point of what I'm showing here is all of these are eukaryotic organisms. They're complex organisms that build all these structures that interact with light. And structural colour is really noticeable. So it's not surprising that people have been working on it now since about the 17th century. So Hooke, Newton, people like that all observed it in the peacock, in flies, in other organisms, and wrote about it and tried to work out the mechanisms. And they already knew that it wasn't like a pigment, a dye, a conventional paint. So people have been working on this stuff at the level of optics for centuries. We'll talk maybe about why structural colour may have some advantages over pigments later, but for now, we've known about it for a long time in some shape or form. And it's a little difficult to explain this over the internet because normally this involves me waving my hands around and miming bacteria and stuff. But it's an interference effect. And what is really important is you have very ordered structures and basically an alternation of refractive indices. So, for example, in the carapace of a beetle, you have different layers. And these different layers have a different optical properties. And the spacing and the width of these layers relative to the light that's interacting them is super important to what you see coming off them. And what you see coming off them in the reflected light can be very intense, very angle dependent colors. You can also have things structural white and structural black, and that's a little bit different. Structural white is super light scattering, and structural black, super light absorption, and they're related effects, and they also exist in nature. So you can do just about anything, any color with structural color. You can also have it work outside the visual spectrum. So it can work in the ultraviolet or the infrared, given the right kind of setup. So it's a very different thing from pigmentation. And then kind of half the reason we got into this is it's kind of a weird thing because in this sort of modern era of genomics, there is almost no phenomenon in nature which we don't know a lot of the genes in the pathways. We don't have bioinformatics. I mean, we have bioinformatics, we have genomics, proteomics, all of those things. It's an odd exception that something that is quite so amazingly noticeable doesn't have a lot of genetics. And for example, one case is the genome of the peacock was published about 5 years ago, something like that. You can see in the title, they talk about the glittering bird. And this is a reference to the structural color in the peacock feathers. And you read the paper and you don't find a single gene they explain to be involved in this. I mean, obviously genes for making feathers are involved. There is also a dark background pigment that makes the color more obvious, but they don't know. And that's really surprising, really odd. And I would say possibly the reason for this is structural color tends not to exist in the model organisms that we studied in the past, the E. coli's, the fruit flies, the nematodes, all of those things which have been the workhorses of genetics in the past. But now we go into an era of CRISPR where we and genomics, where we kind of feel we can take on anything. And I think that's part of the reason that maybe things are starting to shift here and structural color genetics is starting to become a thing, basically. Okay, so the second part of this is how a small company started up based around this idea. So we're in Utrecht in the Netherlands, and if this presentation is a little bit eccentric, it's because it's 7 o'clock at night and the lab were at the Utrecht Bock Beer Festival this afternoon. But the organism that we're kind of interested in now is a bacterium, and we originally isolated what we work on from near Rotterdam Harbour. Quite accidentally, we were trying for something else completely. The important thing to know about it is that it's not a really completely marine environment, and it's not completely a freshwater environment. It's kind of somewhere in between. And the picture to your right is a colony that we got, and it's not the primary colony on the first plate we isolated it from, but it's the immediate respotting, restreaking of it onto a new plate. So it's quite early on in the isolation of it. And it's got some properties which are very typical of structural colour. It's vivid. The colour is amazingly strong, and we'll show later that it's very angle dependent. So if I was to move the Petri dish, you'd see very different colours or no colour at all. It's very, very highly saturated colour. That is completely unaltered photograph. It really is that saturated. And the other thing you might find a little bit strange is that the background, it's growing on an agar Petri dish, is so black. And that's because we use the same trick as the feather of the peacock. We put black ink in the Petri dish, and basically that adds contrast. And that's the trick for finding structurally coloured bacteria. And I know you're already having some success of bugs at doing that. So, but basically, when you do this, lots and lots becomes visible that you couldn't see before. And one of the reasons we became interested in this was it wasn't just that the colony looked kind of cool. And the person who actually isolated this was a medical microbiologist, and she just sort of said, I've just never seen this before. Actually, people have seen it before, but she'd worked 20 years in microbiology and she'd never seen it before. But what we found was kind of interesting is we take the colony and we take a big loop full of it, like you tend to do when you restreak. I mean, this is more than you'd normally want. And you've mixed up the colony, and you see that the colour vanishes. And that's exactly what you expect for structural colour, and it's one of the best tests of it because it's dependent on the cells being ordered. And that's a good way of telling the difference between a structurally coloured colony and a pigmented colony. The next thing we did was we just restreaked it, and we got the picture on the right, which was this amazing striped effect. And it's kind of interesting, and it's kind of deceptive. What you're seeing is the light that's being reflected from certain parts of the restreaked bacteria, and the dark areas actually are every bit as bright as the light areas, but you have to change your angle of viewing to see that. So it's to do with the way the cells are aligned. I'll show a picture of how the cells look underneath this. But it was kind of what really we thought was so good about this was how fast this happened. That there was reordering going on. This was kind of a self-ordering system, and it was happening over 10, 20 minutes. We could pretty much see it, and we've got movies of this reforming. So we thought that's really interesting that this organization capacity is so amazingly strong underneath this color. And of course, we're not the first people to have seen this. If you go back to 1941, unfortunately, the strains have been lost, but one of the really classic microbiologists, Stanier, described something that was almost certainly structurally colored. And it's what was really nice and interesting about the people in that period is it seems to me that microbiology wasn't really taught as microbiology, and the people who were the scientists at the time really were good at physics and chemistry and stuff like that as well. They weren't kind of super specialized, and they seemed to get what they were seeing, if anything, better than a modern audience. And so what Stanier says isn't completely correct, but he couldn't have been correct. The full optics of this weren't known at the time. But he was referencing a debate about, at the time, people could see bacteria really well, but seeing them inside colonies was really still a specialist thing, quite difficult to do. And if you go back to the work of Piper in the 1920s, there were already debates about how bacteria organized into colonies from the way that light formed, the way they formed diffraction gratings and other optical structures. So there was this really vigorous debate a century ago about what was going on, and it was really well informed. Some of these papers back then are fantastic. But there's also some other groups that have seen this. For example, there's a group in France with some really great work by Betty Kientz, who've also been into this, and also some work in Germany. And if you go to the Woods Hole courses for microbial ecology, the students publish their work every year. And if you look into their reports, they're often seeing this kind of thing too. So, but the thing that really made it interesting to us is we accidentally killed the bacteria doing something else. And we realized that even though the bacteria are dead, you keep the color. Well, you have to do it carefully, but this is a structural effect, and as long as you kill the bacteria and keep the order of the bacteria in the colony correct, then you keep the color. And if you can do that, you can make stuff from it. You can maybe make paints or cosmetics or other things. So that's kind of where this starts to become a bit more commercial. So we started a company, and Peter's already explained where the company name comes on. You might have to be of a certain age to realize who we stole the logo from, but it's an adaptation of a prism picture, and with the optics sort of a little bit altered to suit the way that bacteria deal with light, basically. So the next part is what we call IR1, which was this bacterium that I showed before. We kind of do several different things, and I'm not going to talk about all of them, but one of them is generally we do biodiscovery. So we're looking for lots of other structurally colored bacteria, and there's a lot of diversity and interest out there. We're not going to talk about that so much this time. There's also a lot of genomics, which I am going to at least mention. But in order to work on structural color, as soon as we realized there was basically no genetics, we wanted to develop one of these bacteria as a genetic model system. And we chose one that was extremely bright, and it was a flavor bacteria. And there's some reasons to think that you can do transposon mutagenesis, CRISPR, stuff like that in this bacterium. So that's why this one was chosen. So it's a flavor bacterium. This is from phylum, and I keep getting the name wrong because they've changed it recently, but Bacteriodota, Bacteriodete, something like that these days. The thing is, it's kind of a rod-shaped bacterium, and the way it moves is it's almost like a little bit of the caterpillar tracks on a I don't know, a digging machine or something like that, or a tank. There's proteins on the outside of the cell, and they move over the surface of the cell, pushed by molecular motors that kind of cross from the inner membrane to the outer membrane. And these proteins move in one direction, and they push the bacterium over a surface in the opposite direction. As the bacteria move, they kind of corkscrew. So they rotate as they move forwards. And this is a really unusual form of motility that's specific to this phylum. It's called gliding motility. And unfortunately, there's another type of gliding motility that doesn't kind of work the same way, but that's just confusion in the literature. But basically, there's something a little bit unusual here in terms of the mechanics. And for those of you who know about flagellar motility and stuff like that, it's nothing like that. Or, well, the proteins are nothing like that. Maybe some of the overall way the molecular motors work might be a bit similar. But this is the bacterium. And so I wanted to show some pictures of the individual bacteria moving. It's a bit pixelated, but there's a couple of things to notice about it. One of which is that the bacteria, when you normally talk about bacteria, you think, well, the cell wall and it's quite rigid. If you look carefully at these bacteria moving, they're kind of like bendy buses. They actually aren't that rigid. And we think, but we absolutely can't prove this, that has something to do with the way they pack so perfectly together to make these photonic crystal. We really don't know. There's huge arguments raging between a bunch of scientists about exactly what they're doing here. A lot of the molecular detail is still not known. But so what they form is what we call a photonic crystal. And the crystal, you might think of something like sodium chloride. So you have sodium and chloride at different positions. But this is on a much bigger scale. And on this scale is the bacteria. Each cell is forming kind of a different position in the crystal. So for them to boost the color, you've got to have a lot more of them than hit this. They've got to be stacked up in layers. And then you can see something visible. So I don't want to go into too much detail here. We can go into the photonics later if you want. But basically, the color is a side effect of them being absolutely insanely ordered. Much more ordered, much more aligned, much more perfectly positioned than an E. coli colony, for example. Although even a bacterium like E. coli does have some order in it. It's not a completely chaotic mess. But it's to do with the width of the cells and the gaps between the cells. And there's all of these layers. So you're seeing from above fairly highly ordered colony by scanning electron microscopy. And then what is at least approximately hexagonal cross section when you're looking through the colony by transmission electron microscopy. And it's this kind of hexagonal cross section that's important in producing the color. And briefly to say how this is done, what you do when you want to look at the optics of this is you get a machine called a goniometer. And what the goniometer does is effectively it shines light on the sample from every possible angle in a hemisphere. It has a detector that also moves, so you get every possible combination of reflected light in every possible combination of angles, but also you scan through the whole spectrum. So you basically capture this very detailed, very complex data set. And then you combine this with the information on the structure of the bacteria, which you basically measure from electron microscopy samples. And you combine it into a mathematical model. And that's how you kind of solve it. It's not quite like solving a protein crystal structure, but it's the nearest analogy I can get. So it's a very mathematically intense process. And thank goodness we don't have to do it ourselves. We collaborate with Silvia Vignolini's group, and she's an optical physicist. And basically she translates the color into structures for us. I just wanted to say one thing more about form and function. Because this is all about geometry and alignment and spacing, if something produces a similar optical effect, it may well look quite similar too. So this is part of a peacock feather to the left. It's been a bit messed with. People have removed some structures because they wanted to image a particular bit of it. So not all pictures of a peacock feather look like this. And then to the right, structurally colored bacteria. And it is no surprise they look the same. It doesn't mean that the peacock feather is composed of bacteria, but that the optical function is remarkably and amazingly the same. And I would say that the peacock feather is actually very close to what the bacteria are doing. It's kind of peacock feather on a Petri dish. And I only won't say that if there's any optical physicists in the audience, because they've got a few objections to that, but it's as close as I can get, basically. What does it do is a whole can of worms. We don't know is the simplest answer, but as people always ask this, there's only one thing I can contribute. And it goes back to what I said at the start, that's maybe color that you see isn't always actually the function of what you're seeing. So we did an experiment where we put IR1, quite by accident again, we've had a lot of luck in this project, next to another bacterium. And we did it on a plate that had almost no nutrients on it. And what we found is under that circumstances, IR1 got aggressive and basically ate the colony next to it. And this, you can see it here. So at position X in panel A, it's kind of moving into the colony next to it. And position Y, what you're seeing at the kind of battlefront between these two strains, it's suddenly forming this really organized structure. And we don't know exactly what's happening there. We do know that if you make a mutant and you knock out structural color, you also lose this ability to attack nearby colonies. So I can't explain it completely. I can just sort of talk about it by analogy and say we think of it like the bacteria are like this sort of Greek phalanx or Roman legion, and they're kind of super organized, and they charge into this mass of barbarian E. coli that are sort of inferior at sort of bacteria to bacteria combat. We only know part of the story here, but one of the other things that's coming out, and we're going to publish this, I hope, early next year, is structurally colored bacteria can be extremely common in parts of the ecosystem where there's absolutely no light. So we're kind of moving towards the idea, but it makes us a bit sad, that it may be that the color is an accident and it's the organization of the bacteria that's really important for its survival value. And however, maybe we can do something with the color. And the other thing is it's fantastic to work on. If this organism wasn't so brightly colored, we'd never have seen all this stuff happening between the two colonies. So it's kind of super convenient at a scientific level. So just to pop back to the stripe, it's basically a product of the way the cells are orientated and the way that you're looking. So if you look at viewpoint 1, as I say, you've got a sort of, the dark areas are not dark, they're just you're looking at the wrong angle, and you change the illumination or you change the viewpoint and your picture will change. This means when you look at structural color, you've really got to change the angle a lot. And this kind of makes sense in nature. In the butterfly or in the peacock and things like that, the organisms that look at each other, they're flying or they're doing a mating display or something like that, and they're changing angle. So it becomes super, super noticeable. So if you're looking at structural color on your Petri dish, just kind of keep shifting the angle, shift the light, and suddenly you see something. We've had situations where we thought we've had a mutant that isn't structurally colored. We've thrown it away, and the light from this window has come into the microbial waste disposal bin, which I guess we shouldn't have had open, but we did. And you see suddenly the color, but it's color from a different angle than you were expecting it. So yeah, it's kind of cool. But to move from the way the bacteria behave, one of the things we did is we talked to some art students and some design students. And this is particularly Elvin Karana's group. She's at Delft. And I think she's done some things that may interact very well with bugs. Some of her students have basically explored what you can do in terms of the design potential of living structural color. Preserving it and making paints from it is another issue, but she's mostly focused on the living stuff. She's got a very nice imaging setup where she's making movies of the development of structural color. I think Peter mentioned this, that you were kind of interested in this. There is something, though, that she's not doing that I think would be very cool to do, and that is she's only got a single camera. Where structural color gets really interesting would be to make movies from multiple viewpoints with multiple cameras simultaneously, and I think you'd learn a super amount by that, and it's something that you just can't see as an individual. But she's exploring that. And also, if you look at her work, she's also made this rather cool digital tool where you can actually kind of simulate structural color on an iPad or a computer. You can use it as a sort of design tool. So the color's noticeable and it's being used within that sort of design area. And then go to synthetic biology and what, however cool structural color is now, it isn't really completely ready to be products, to be paints, to be cosmetics. We can discuss why later, but partly because of the great deficiency in the genetics, and partly because however good it is, we want to improve it. We started to get into synthetic biology and genomics and genetics and all that level of stuff. So we think that, I mean, we're kind of militantly pro genetically modified organisms around here. I've had quite a lot of arguments with people over the value of it, but I think what I would say is if it comes to a contest between sustainability and the possible dangers of GMO, then sustainability and climate change is a much bigger threat. This isn't to say we should ignore the effect of GMO organisms, but relatively speaking, I would go with GMO every time if it makes our environment better. That's kind of maybe soapboxing on a general point, but specifically, what can we do with structural color? So just to go back to my point, it is weird and amazing how little there's been in terms of genetics. There have been a couple of interesting papers in butterflies. There was one group that did a CRISPR knockout, and they were trying to knock out the pigment of the butterfly wing, there's quite a lot known about that. And they accidentally hit a structural color, a gene that basically affected both pigment and structural color. But it's difficult to do that in high throughput in the butterfly. And of course, bacteria are just super, super, super easy compared. So almost always you'll hit, the bacterial stuff will hit maybe 5 years ahead of people doing it and other things. But the reason we want the reason we want synthetic biology completely is at the moment, making this color happens on Petri dishes. It happens on plates. If we're going to make this commercially, we're going to have to get it into fermenters, and we're going to have to change the way the organism grows to do that. That's happened in the past. I mean, things like penicillin, other industrial processes often first are seen on a Petri dish and then they're adapted to upscaling. But we think that has to happen for this to be useful. And of course, you need to drive down costs. You may want the organisms to grow on materials which are very sustainable. And of course, we want to actually understand the genetics, but we want to do all of these things, both for commercial reasons and also for scientific academic reasons. So initially, we got a transposon system that had been used in other flavor bacteria. So this is something that jumps around the genome, it disrupts genes. And basically, we looked for, it makes mutants, and we looked for mutants that were either what we call dull mutants that were knocked down in structural color, or occasionally color shift mutants, which are particularly interesting. And in fact, I think what we sent you, we sent you a couple of color shifted mutants as well as the wild type strain. And then you just sequence the genome, find out where the changes are. You relate these changes to the structure, and that means doing electron microscopy. And then the bit at the bottom, the M to P, is basically what you see from goniometry. It's how they see the kind of optical analysis. And you put all this stuff together and you make basically the first really good linkage between a genetics and structure, initially using this mutagenesis method. So some of this is published, and there are things like the really detailed structures involved, and a lot of the genetics and the strains. I think some of you have seen that. So we have already established some of the genes from this process, also links between nutrition and color, how you grow the organisms, how to grow the bacteria, and how you can change the color by doing that. And then we live in the era of CRISPR. And so, of course, CRISPR, we can go the other way. Rather than looking for bacteria that change color and then going and sequencing and finding the genes which are knocked out to change those color, we can now start to take individual genes and say, well, is that involved in structural color or not? And then we can use the CRISPR gene editing system to do that. And many thanks there to Raymond Stahls, who's as kind of quite a well-known CRISPR group at Wageningen University in the Netherlands. They helped us build the tools to let us do this more specific genetic engineering for us. But so this is possible. Yeah. Okay. So, I mean, we're really not We're only a few cycles through this. But what we're trying to do is a sort of reiterative process where first we use both proteomics and genomics to see what we think is going on with the pathways involved in structural color. We modify some genes, find the ones that do interesting things. We demonstrate this with an optical structure with our collaborators, and then we try to convert the optical structures into materials. So if we shift the organism's colonies to becoming red, does it also become a red material after you kill the organisms and you preserve it and try to make it in some sort of paint or something like that? And once you learn that, you go back into this cycle again. And this is taking a while. We've probably gone round this loop, I don't know, 1.5 times or something like that, or maybe 2 times, but it's not something that we've cranked a huge number of times yet. But we are getting going, and we're kind of gathering speed each time. We learn a lot each time we go round this cycle about what the pathways are involved. This is actually quite an old example, but I'll give it because it's the cleanest example. For example, we have a gene which we call HYPE16. That's because we haven't really in detail a clue what it does. It's not a gene with any really obvious known function when you look in the database. And that's quite common in structural color, and that's nice. That's interesting that there's new stuff here. And you can see that basically in both cases, basically when you knock this gene out, which is moving from A, which is the wild type, which is predominantly a green structural color, to B, which is red shifted, that you see all of the changes, what I said before. So you're seeing changes in the cell size and cell organization, but they're still very organized in the mutant, the red mutant. And of course they'd have to be. If they were disorganized, you'd lose the color. And that's kind of what we're trying to crank at a rather bigger throughput at the moment. So you've got all these really nice bacteria, but kind of what use is it? I already said that you can make material from it. They don't have to be alive. And there's a good, really interesting movement, which I'm sure you're part of, which is using microbes as materials. A lot of the examples we see around here, and some of these designers, they're from Belgium. I think, yeah, Maurizio's definitely Italian, and there's some Dutch designers I know. They're going really heavily into mycelia, into fungal systems, which make quite interesting structural materials. Because of the way you can dry down the mycelia, you can grow them well on substrates that are quite cheap, quite reusable. So they're making stuff out of these things, but they really don't look very pretty. You also can use fungi to extract pigments, and that's also been done with bacteria, but there's this kind of movement emerging. So what I said before, what you've got to do, if you want to make a material, you've got to start with the cells alive, and they've got to be ordered, they've got to be aligned in the right kind of way, make this photonic crystal, and you've just got to stabilize them. I'm not going to go too much into how we do this, but if you could just take for the moment, there's a preservation method. And this is kind of what you do at the moment. It's very inefficient. You grow them on agar. You basically preserve them on the agar. You lift the flakes off the agar, which are now dead, but structurally colored. Sometimes the flakes don't stay flat, in which case you get actually quite a variety of colors coming off it. And you can also, but you've got to be quite careful about how you do it. You can grind these flakes into smaller particles, in which case you can still keep some color, but it doesn't look nearly as orientated or as glittery or as reflective. It looks much more like conventional paint when you do this, when you really take the, and of course, if you grind them down too far, you lose the color completely. But you can do some quite nice things. So everything in this picture is dead. It is hydrated. So panel A is a basically it's a cellular phagolytica, so it's flavobacterium. It's not IL-1, it's something else. But it's streaked in a way that it makes it particularly homogeneous color on an agar plate, but then it's dead on that plate. B, the top and the lower part of this is really fun. This is what happens, and it's not easy to do. If you float this film that you see in A off the plate. So what you're seeing in B is the film is floating on water. Underneath the film is a, I can't remember what it is, 25 euro cents, something like that, but it's an EU coin. And you can see that it's transparent. If you look at it from a different angle, you look at the bottom panel in B, and it's reflective. So you've actually created a material which, by the way, by how you've orientated the organism, it reflects light in a very specific, very selective way. So it's quite a sophisticated thing. And then C was us playing about with fabrics and trying to fix the material onto fabrics to make basically a form of textile dye. So these are all materials. And you can have a lot of fun with this, as some of the designers we work with have done so. So they've made sort of flakes and spheres and, yeah, all sorts of interesting stuff. And there is one other process, and here again, sorry, because of possible patenting, I'm not going to say exactly how we did it, but there are even ways which you can take the dead flakes and chemically treat them and blue shift them. So you don't have to necessarily stay with the color that you start with. You can actually do some later downstream chemical modifications and change the color a bit too. So some possibilities. We are a company. We do very much have the very big problem that this is a long way from the market. And whilst big paint companies and cosmetic companies, people like that are quite aware of what we're doing. We've done some work, for example, L'Oreal, the cosmetics company. They want to see many things from us before they really would take this on for a mass market. And particularly the scalability is the key question here. But cosmetics, there's some really interesting possibilities as colorants. You can say, these are bacteria, but actually they weren't too worried by that. And as long as with the cosmetics industry, they don't like GMOs so much, but they are incredibly interested also not just in colors, but whether any of this can function in UV protection. Paints, very interested in colors. They don't want all of this glittery, shiny effect, except for specialist applications. But they do want to replace some of their pigments with more sustainable alternatives. And it's a different subject, but they really, really, really want to get rid of titanium dioxide acting as an opacity and whitening agents, and whether bacteria or other biological materials can act as whiteners is an interesting subject. There are some cellulose things coming through that are really, really cool. Textiles is difficult, but we have also talked with textile companies, and I think we're filing a grant with some Portuguese textile people, and we are getting approaches from painting it, but early days. But that's kind of actually what we do. We develop stuff for these markets. I hope that was kind of together. I guess we'll find out from the questions, but broadly speaking, the first message is you can actually really create biomaterials from bacteria that are structurally colored, even if at the moment creating large amounts is difficult, and that's what we do. We think that there are possibilities in terms of performance, the fact that they're bio-based, sustainable. Flexibility is an interesting one because you only have to change the distribution, the ordering of the bacteria a little bit, and it changes the color. So in principle, you can make any color from this. And also possibly if you can scale it, bacteria are not expensive per kilo, at least in a fermenter. So maybe you can even undercut conventional dyes. But again, a lot of this has to be proven. We are applying this to make production strains, but also we're going very deep into the biology of this, which is starting to move really quickly. So I'm not sure how much more there is. There's only a couple of slides. We've got a lot of really good collaborators, notably Sylvia, who does the optics, Raymond Stiles, who basically helped us with genetics, and Elvin Karana, who is involved in the industry and the design side. And then there's also some projects which go into proteomics and genomics, which I haven't talked about here, but there's also some professors in Germany and Portugal and Sweden who work with us. And that's a really good thing because we're basically almost nothing as a company. There's only a few of us. 3 of us makes for a sort of big lab meeting. So yeah, we've got a way to go, but thank you for your attention. And then I've just got one slide to go, which is that one of our students, Alvaro, got bored and resuspended some of his bacteria in a I think it was in a transparent shower gel, and then he got it to rotate, and he made a whole series of really cool movies based around it to show what the colors can do. And what you can see, I talked about earlier, this sort of patterning, this striped effect, and you can see some of the striped effect in what he did. So I think I'll leave it there. Thank you very much for your attention.
Thank you. Thank you very much. Can you hear me?
Yes, can.
Yeah. I'm sure we have some questions. So if anybody has a question for Dr. Ingham, I could, if you just raise your hand and ask the question, and if he can't hear you, I can translate it. And maybe I can start with a question. Very early on, you talked about how the bacteria can also split or can refract other wavelengths of light, like in the UV. And I think at the end you mentioned something about UV protection. Is there anybody, like, first of all, is it the same bacteria that would do it would be able to split UV lights at the UV wavelengths? And how would you actually discover those bacteria? Are you actually looking into that?
So yeah, the thing is, basically, to put it simply, the color that you see is very dependent on the width of the bacteria. So the narrower the bacteria, the more you go into the blue. And if the bacteria are really narrow, then you're just about pushing into the UV part of the spectrum. So one way of looking is to look for just a spacing of the bacteria that's kind of super fine, super small. The reason we noticed this is that when Sylvia's group did the optics, did the goniometry, they do the full spectrum. So they noticed then that there was activity both in the UV and the infrared, as well as in the visible spectrum in the same bacterium. That leads to a bit of a mystery because we're not sure what the structures are, what the periodicity is. There must be a periodicity on a really fine scale to explain the UV reflectivity. But the way you look is you use a UV camera, and you can actually see it. So we bought a load of UV lamps from a nail salon, just sort of long range UV, and you can actually shine it on the colonies, and then you can use a UV camera to see it. And you can actually build one of these cameras yourself. You can actually take a broad spectrum normal digital SLR, but some makes are much better than others. And you've got to mess around with the filters, and you can actually create one that can pick up ultraviolet.
But first, you would want to see that the bacteria has to be packed very tightly to be able to achieve that at the high wavelength.
Yeah, I think though that what I would do probably is you would just take your colonies, like you're taking the colonies that you're looking for now, and you would try to set up a UV camera screening setup, and you would just pick the ones that look really reflective under UV first, and then look at them and see if they fit the other criteria later.
Got it. Are there questions on the line? Just unmute yourself if you have a question for Dr. Ingham. And any questions? I think we have a question in the room.
Just one question in terms of length of time for developing. So he's talking about UV and as a protectant or cosmetics. how far away do you see us being from getting there?
Did you hear that?
Yes, yeah, basically how far from the market. The answer to these questions is almost always how much money. I mean, it's really true. I mean, we've done this over quite a long period of time, but we've not spent a lot of money doing it. I think if you took, for example, a serious synthetic biology company, I don't know, Ginkgo or whoever, and they put a couple of million into proving the strains, and a big paint company put into the effort in the processing of the strains to make the pigment, I think it might take 5 years or a little bit longer than that. The barrier really, though, is that understandably, these big companies don't want to push their whole production over to a weird technology very easily.
I have a there's a question here. Okay, what's your question?
You said that can you change the color of the bacteria? Can the bacteria lose the chemicals, the color, can the color dim through osmosis or something over time, over generations?
Did you hear that?
No, sorry, could you repeat it, please?
Oh, can I repeat it for you? I think he was asking that over time, can the material lose its color once it's been fixed? Is that what you're asking?
Yeah, over generations.
Over generations.
Right. So, but over generations, do you mean alive or dead? Whose generations? Over people's generations or
Material.
Bacteria.
Okay. So first take it on the plates, how they behave there. They're kind of weird because if you grow them on a really rich medium, they make lots of structural color and it's really nice looking, and they die off really quite quickly. And as they die off, they become disordered. And so in the center of an expanding colony, you'll start to see them go dark and black, and that's when they're dying and they're losing the color. Sometimes they can age in more interesting ways and you get kind of blue shifts happening. So you can see a sort of slightly more beautiful aging. And so this can happen over a period of a day, weeks, something like that. If you grow them on low nutrient medium, the color may seem less, but for some reason it's a lot more stable. So the nutrients affect the way this stuff behaves. If you make a hydrated preserved material, then if you stop it dehydrating, the color will stay almost indefinitely. At least, I mean, the company's about 5 years old and we've got some samples that are still colored. If the samples dry out in an uneven way, you'll lose the color. And then the last bit of this is there are ways, and we did this again by accident, if you preserve them for electron microscopy, where you use the process of critical point drying, where you take the water out, you can sometimes, if you're very lucky, leave the color. It distorts, but it's still there. So in principle, they can actually be a dried material with an indefinite shelf life, but you've got to be a bit lucky to get it that far.
Thank you. Thank you. So when you're growing bacteria in the lab, is it like tens of liters, hundreds of liters, or you have big fermenters? What's the scale here?
No, we're still Petri dish people mostly. I mean, the problem is the step to the fermenters is a big step, which once you can take it, then it doesn't matter if you do 10 liters or 100 liters, but we're really finding it difficult to make that step. So we're not doing big throughput.
Any other questions?
Any more artists?
One of the things that I thought was really interesting from one of your papers is where you added carbohydrate source from algae, and that stabilized the color. And I thought, because when I was thinking about what's the purpose of this, thinking about where you found the Flavibacter, it's on a surface in a very complicated environment, and maybe so. Do you think that that's another plausible reason for structural color, is to help them survive in this, marine environment where they're stuck on a surface of a clam or something like that, and there's water swishing around them, or predators that are. So the work that you shared with us was about IR1 eating another colony, but it also seems like survival against the environment would be a great reason for structural color.
Yeah, absolutely. No, IR1 seems to eat almost anything when it gets desperate enough. It's so what we know is, firstly, that the genes for using these polysaccharides in algae, if you mutate them, you do affect structural color. So there does seem to be a link there. The genomics people also say that the complex polysaccharide genes are, which are very specific often to specific types of algae, are very important. So basically everything is pointing in that direction, that there's a link between these things. What we don't know, and we also know, we know that you can grow them on seaweed. I mean, we've just bought sushi wrappers, rehydrated them, and you can get structural color on them if you try at it. We don't know really what happens in the environment, though. And we don't really know whether it's the color. We kind of have a suspicion that I already suggested that it may be cooperative in eating other bacteria, but also if they're actually, can you see my hands? Is all this visible? Okay, so the hands of the bacteria, and they're growing on an algae. If they're super chaotic and they degrade the polymers in the algae, they'll float off as sugars, and they'll grab some of them, but other bacteria might get them too. It's possible, if they're super closely packed and super organized, that they kind of grab all the food. They kind of gang up and get a monopoly on degrading that bit of algae. That's a possibility too. And there was another suggestion from Portuguese scientist, and he said, it could also be that they're on the surface of the algae, and because they're so closely packed together, there's capillarity going on between the bacteria, so they don't dry out as much, because you can imagine the algae floating to the surface, it getting the sun. So it's all speculative, we don't know.
Okay, I don't see any other questions. So I want to thank you, Dr. Ingham, for joining us, and also for supporting bugs. We didn't talk about this before, but Dr. Ingham and Hoekmine have shared their research materials, and we're growing some of those strains in the lab right now. And if anybody is interested in working on structural color and other colors in bacteria, talk to Lisa or myself about our chromoproteins group, which group of scientists and non-scientists, some artists that are interested in making reagents that could be used for bioart. And that's actually what we're going to do in the next hour here, when a number of people have joined us, and we'll be drawing with bacteria on agar plates, and that's always a lot of fun. So again, I want to thank you for sharing your Saturday evening with us, and I know you could have been drinking beer, and I know that's a very popular thing in the Netherlands. I hope that we will continue to keep in touch, and we really enjoyed hearing from you.
Thank you very much. It was a great evening, and yeah, we're really, if there are scientists or artists who want to go further with this, we're really happy to help in the future.
Thank you very much. Have a great evening.